Okay, ladies and gentlemen, thanks for the introduction. And also thanks to the OMICS organization for inviting me to present some of the work we are doing in Amsterdam. And it's interesting always to be back in San Francisco, although we did not have much time yet to enjoy the city, but maybe that will come. What I will do is briefly talk about some strategies to optimize the use of bacterial cytochromes P450 for drug discovery and development. And I can tell you that it is in fact work from Harini Venkataraman, and she presented and defended her PhD thesis on December 2nd last week, and it is an illustration indeed of her work, primarily what I am going to talk about. And as probably most of you know, is that the cytochromes P450 are a large number, in fact more than 5,000 highly conserved enzymes occurring in every domain of nature, including in humans. And in humans, the P450s are an important class of phase 1 drug metabolizing enzymes responsible for about 75% of the metabolism of 75% of the metabolism of drugs which are currently on the market. And drug metabolites are relevant for pharmacology and also for toxicology for drugs, and that is one of the reasons that there is a great interest, in fact, in cytochromes P450. There is a large number of cytochromes P450, but I will not go into the details of that now. I just wanted to illustrate that if you compare the human cytochromes P450 as are indicated here, a number of them with typical substrates to characterize these human cytochromes P450. They have a certain catalytic activity, but basically they are a bit lazy compared to, for example, bacterial cytochromes P450. And there is one P450, cytochrome P450 BM3, I'm going to talk about, which is 1700-fold more active than human cytochromes P450. And moreover, the mammalian P450s and also the humans are able to metabolize drug-like compounds, but the disadvantage is that they are not very stable. They are membrane-bound, and moreover, they are not easy to overexpress to a large extent in bacteria. And as I mentioned already, their catalytic activity is rather low. On the other hand, the bacterial P450s are not able, especially not the wild-type BM3, is not able to metabolize drugs. But on the other hand, it is very stable. It is a cytosolic enzyme, which has certain advantages. It is easy to overexpress in bacterial systems, and it has an extremely high catalytic activity. So that was the reason, in fact, to use the high catalytic potential of the cytochrome P450 BM3, but to modify, in fact, the limitation that it is not binding and turning over drugs. And one of the reasons, or one of the possibilities to do so, is going into mutations of this enzyme system. And the brief summary of the BM3 P450 is that it is a soluble P450 from Bacillus megaterium. The reductase domain is fused to the oxygenase domain. It has the highest ever detected activity of a P450. There are several crystal structures available, and the natural substrate, in fact, of substrates are fatty acids, and it is specifically the hydroxylation of these fatty acids which are the most relevant reactions of the BM3. So it has been suggested already quite some time, in fact, using protein engineering in different ways, site-directed, random mutagenesis, saturation mutagenesis, in order to change, in fact, the substrate selectivity of this BM3. And in the meantime, that has been very successful. And if I look, for example, to our own library of BM3 mutants, it is probably a library of 10,000 different P450s, of which 100 to 125 have been extremely well characterized with different substrates and different reactions, which are mimicking, in fact, or covering the chemical space of drug-like compounds. And what we have done in the meantime also is create a kind of platform of cytochrome P450 BM3, which is schematically described here. So apart from mutation and expression, automated selection and picking of colonies of the expressed BM3, there is a characterization of the BM3s with certain substrates, and after the selection of active ones with the appropriate activities, it is possible to upscale. This is not working neither. It is possible to scale up the P450s. For example, in our case, this is not working neither. Let me see. Yeah, this one. Okay. It is to scale up in our laboratory until 3 liters in order to generate higher amounts of metabolites, which can be identified with NMR, mass spectrometry, and be used for affinity screening and so on. And Harini here, in fact, as I mentioned recently, at VU University, in fact, applied, and I will illustrate today 3 samples, BM3 mutants in relation to ionons, testosterone, and the number of NSAIDs. And the structures of these compounds, quite variable in structures, are indicated here. And basically, it is all about regio and stereo selectivity, and secondly, about selective aromatic and aliphatic hydroxylations in these examples. There is quite some more examples, but I'm going to focus, also given the time, only on these 3 examples. And ionons are a class of nor-isoprenoids widely used in flavor and fragrance industry with different, let's say, flavors, for example. And alpha ionon is one of the isoprenoids with organoleptic properties. And it is interesting to see that notably the hydroxylation at the 3 position of the ionons in the 2 different enantiomers of the ionons also are interesting precursors for synthesis of fine chemicals. And our goal was to identify mutants capable of producing the different diastereoisomers, preferably with high stereoselectivity and a high activity. And here is an illustration of the fact that, in fact, looking to the 3 hydroxylation in the trans and the cis configuration, and also secondary oxidation into a 3-oxoionon is indicated to a varying extent in different mutants. And there is a number of mutants which are highly selective towards trans hydroxylation. And there is also a number of mutants which produce, let's say, both the cis and the trans. And if we look further into the details of this, then we can see that in this case, a certain mutant, MT80 mentioned here, and MT33 have been used with the different enantiomers, in fact, of the alpha ionon to produce products. And in fact, all 4 diastereomeric products to a large diastereomeric excess, namely 90 to 95%. And moreover, the turnover of these reactions is very high as well, and it's easily possible to get complete conversion of a substrate, for example, 1 millimolar with minimal secondary metabolism, thus yielding indeed the different diastereoisomeric forms as indicated here in this case. This has led to a publication indicated here, but also to a patent application in collaboration with a company in the Netherlands. So in fact, we have 4 mutants which selectively generate the 3-hydroxylated diastereomeric products of this ionon product. The second example is with testosterone, and indicated here. And the testosterone is an interesting compound because it has a large possibility for hydroxylations, notably with P450s at the A and the D ring, and mostly, let's say, the beta configurations of these hydroxylated products are formed. But our goal was, in fact, to identify mutants with BM3 with altered C16 stereoselectivity, because as yet the 16 alpha hydroxylated product had never been biocatalytically synthesized before. This has been rationalized with computational modeling, including molecular dynamics and a generation of models of the mutants, but for the sake of time I cannot go into the detail of that at this moment, and I would like to continue by illustrating that if we look to the 2 beta, the 16 beta and the 15 beta products, then we can see here in different color codes that in fact different hydroxylated testosterone metabolites are generated. But interestingly, there were 2 mutants with here in orange indicating a new metabolite, and by HPLC and also by NMR and advanced NMR structures, we were able to identify the structure of this new metabolite, and it appeared to be the 16 alpha hydroxylated testosterone based on the analysis of the product with the different techniques shown here. And here you can see that, for example, there is a mutant which is primarily producing the 16 beta hydroxy, and there is another mutant which is primarily generating the 16 alpha hydroxy compound, in addition to some other products which can chromatographically easily be separated, in fact. And here it is summarized, in fact, about the C16 hydroxylation. One isomer is primarily given the 16 beta, and another one is primarily giving the 16 alpha. So this one is 0% in this ratio of the 2, and this one is 25% stereoselective for the 16 alpha testosterone. This has been rationalized, and it has been proven and seen that the serine at position 72, which is in the entrance channel, in fact, is important for anchoring the steroid in such a way that if you replace the serine at this position by a hydrophobic isoleucine, the stereochemistry of the 16 beta is changed to a high extent to the 16 alpha. There is some other mutants as well which have been mutated, and it's interesting to see that when we introduce the serine 72 isoleucine mutation in another BM3 mutant, in this case it is the M11, for example, then you in fact change also in the other mutant the stereoselectivity from the beta into the alpha configuration. A strong indication indeed that is a very selective mutation which is leading to a difference in the active site and in the orientation of a steroid like testosterone, and giving with large enantiomeric excess indeed either the 16 beta or the 16 alpha hydroxylated compound. The last example I want to show is phenamic acid, or NSAIDs, non-steroidal anti-inflammatory drugs with the general structure indicated here. That's phenamic acid, and they are often used in migraine, for example. They are known to be hepatotoxic, unpredictable based on preclinical studies, which is idiosyncratic, and they are also nephrotoxic compounds. And here is 3 examples: meclofenamic acid, mefenamic acid, and tolfenamic acid. And these 3 examples have been used in order to ask the question whether, in fact, the human relevant metabolites of these drugs could selectively and to a high extent produced by the BM3 mutants as indicated here. And for these NSAIDs, for example, the human and rat liver microsomes containing P450s only produce low amounts. Moreover, it is important to have metabolites produced according to regulations by FDA and the EMA, for example. Metabolites in these days also have to be included into the regulatory programs, and for that purpose you need sufficient amounts of selective metabolites indicated here. And in fact, the approach is the same with a number of BM3 mutants, the production of different hydroxylated metabolites, and it is here the 4' hydroxy, the upper one, the 5 hydroxy, and the 3' hydroxy, and according to color coding, they are discriminated here. And it is clear that in, for example, mefenamic acid, the 3', the 4', and the 5 hydroxy compounds are to a certain extent selectively produced by different mutants at high turnover numbers, which is really sufficient for the production of, for example, in this case, about 7 milligrams in 50 milliliters of incubation, and the same is true for the tolfenamic acid, also illustrating selectivity over there. So basically the applications, to summarize this third example, is that the BM3 can be used in the first step of the oxidation in order to yield an aromatic hydroxylation, but the BM3 is also used in a second step, generating a quinonimine, which is a chemically reactive intermediate, possibly responsible for the hepatotoxicity of these compounds. And they are also conjugated to glutathione with the add of glutathione as transferases. And this strategy is useful to generate a reactive intermediate and to trap it with GSH conjugates for identification and protection. And the second application is that it also has been used in a multi-component reaction in which in one cellular system we have combined BM3 and co-expressed it with human glutathione as transferases in order to generate in a cellular system 2 reactions sequentially in order to generate the products which are important for the toxicological risk assessment. So overall, and in conclusion, I think I showed that BM3 mutants can be highly regio and stereoselective. In this case, in hydroxylations, I did not show other reactions, but they can do that as well. Complete conversion can be achieved in some cases with minimal over-oxidation. Turnover numbers of 4,000 and more give good perspectives indeed for large-scale applications, and high expression levels combined with the total turnover numbers are also good for, let's say, cellular catalysis for biocatalysis purposes. And the challenges and the issues, in fact, for the future are to increase the stability, solvent and temperature stability, to amend the cofactor regeneration in order to make it cheaper and even more efficient, and also to alter the specificity and the selectivity of these enzymes dedicated for specific purposes. And the last slide is in fact indicating the group, and in this case Harini, let's say, was essential, and staff member Jan Commandeur, and also of course several sponsors which have been contributing over the years to our groups. And I would like to thank you for your attention.
Thank you very much. If we could change the slides over while we just take time for one question, I think, if anybody has any questions.
Bacterial P450s can use hydrogen peroxide as an oxidant.
Yeah.
Have you tried this as well here, or is it NADPH?
BM3 is exactly doing the same as all P450s do indeed, and they are also peroxygenases and use hydrogen peroxide and other peroxides as an oxime donor for reactions in P450, and the BM3 indeed is doing that as well. Yeah.
So your reactions were with NADPH?
Our reactions indeed are the monooxygenase reactions in which we are using NADPH. But in principle, the oxine donors via peroxides are also capable for BM3 catalysis. And we have used that, but with an NADPH regenerating system, it is no problem in order to use reasonably cheap cofactor systems. But cumene, hydroperoxide, hydrogen peroxide, and other peroxides are also oxine donors for P450s.